Respiration in plants. 1. The breakdown of glucose to pyruvic acid is called glycolysis. 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 2. Glycolysis was given by Gustav Emden, Otto Meyerhof, and J. Pernas. Gustav Emden, Otto Meyerhof, and J. Pernas. Gustav Emden, Otto Meyerhof, and J. Pernas. 3. Glycolysis is also called pathway. EMP. 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 4. First step of glycolysis is catalyzed by enzyme hexakinase. 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 5. No of reactions in glycolysis are 10 10 10 6. Net ATP and NADPH produced in glycolysis per 4 glucose molecules are 8 ATP, 0. NADPH, 8 NAD will be produced, not NADPH. 8 ATP, 0. NADPH, 8 NAD will be produced, not NADPH. 8 ATP, 0. NADPH, 8 NAD will be produced, not NADPH. 7. Write all the reactions of glycolysis. A. 8. Write the enzyme involved in glycolysis. Write the enzyme involved in glycolysis. 9. What are the main cofactors involved in glycolysis? Mg2+, Mg2+, Mg2+. 10. Reaction in glycolysis at which NAD plus H plus is released is GLYCERALDEHYDE3 phosphate to 1, 3 bisphosphoglyceric acid, GLYCERALDEHYDE3 phosphate to 1, 3 bisphosphoglyceric acid, GLYCERALDEHYDE3 phosphate to 1, 3 bisphosphoglyceric acid. 11. Substrate level phosphorylation occurs at which steps? 1. 3 bisphosphoglyceric acid to 3 phosphoglyceric acid. 1. 3 bisphosphoglyceric acid to 3 phosphoglyceric acid. 1. 3 bisphosphoglyceric acid to 3 phosphoglyceric acid. 12. Pacemaker enzyme in EMP pathway is phosphofructokinase, phosphofructokinase, phosphofructokinase. 13. Rate limiting step in EMP pathway is fructose 6-phosphate to fructose 1,6 bisphosphate. Fructose 6-phosphate to fructose 1,6 bisphosphate. Fructose 6-phosphate to fructose 1,6 bisphosphate. 14. Consumption of ATP occurs at which steps? Glucose to GLUCOSE 6-phosphate and fructose 6-phosphate to fructose 1,6 bisphosphate. Glucose to GLUCOSE 6-phosphate and fructose 6-phosphate to fructose 1,6 bisphosphate. Glucose to GLUCOSE 6-phosphate and fructose 6-phosphate to fructose 1,6 bisphosphate. 15. What are the three fates of pyruvic acid produced? I. Alcoholic fermentation. E. Lactic acid fermentation. E. Aerobic respiration. I. Alcoholic fermentation. E. Lactic acid fermentation. E. Aerobic respiration. I. Alcoholic fermentation. E. Lactic acid fermentation. E. Aerobic respiration. 16. Yeast converts pyruvic acid to and CO2 and ethanol. CO2 and ethanol. CO2 and ethanol. CO2 and ethanol. 17. Write the two reactions and also the enzymes involved in these reactions. 18. Pyruvic acid is reduced to lactic acid by enzyme lactate dehydrogenase. Lactate dehydrogenase. Lactate dehydrogenase. 19. In both lactic acid and alcohol fermentation, less than % of the total energy is released. 7%. 7%. 7%. 20. Yeast poison themselves to death at % of alcohol. 13. 13. 13. Aerobic respiration, TCAN. It's 21. In which reaction pyruvate dehydrogenase is used and where is it found? It is used for converting pyruvic acid to acetyl-CoA. It is found in mitochondria. It is used for converting pyruvic acid to acetyl-CoA. It is found in mitochondria. It is used for converting pyruvic acid to acetyl-CoA. It is found in mitochondria. 22. Write the linking reaction. Pyruvic acid plus CoA plus NAD plus pyruvate decarboxylase acetyl-CoA plus CO2 plus NAD plus H plus. Pyruvic acid plus CoA plus NAD plus pyruvate decarboxylase acetyl-CoA plus CO2 plus NAD plus H plus. Pyruvic acid plus CoA plus NAD plus pyruvate decarboxylase acetyl-CoA plus CO2 plus NAD plus H plus. 23. 2. Coenzymes and cofactor required by pyruvate dehydrogenases. Dash. NAD plus. Coenzyme ANMG2 plus. NAD plus. Coenzyme ANMG2 plus. NAD plus. Coenzyme ANMG2 plus. 24. Pyruvate after entering to mitochondrial matrix undergo dash, oxidative decarboxylation, oxidative decarboxylation, oxidative decarboxylation. 25. How many NAD are produced by 2 molecules of pyruvic acid during oxidative decarboxylation? 2 molecules of NAD. Dash. 2 molecules of NAD. Dash. 2 molecules of NAD. 26. TCA cycle starts with the condensation of dash with dash and water to yield dash. Acetyl-CoA, OAA, citric acid. Acetyl-CoA, OAA, citric acid. Acetyl-CoA, OAA, citric acid. 27. First step of TCA is catalyzed by citrate synthase, citrate synthase, citrate synthase. 28. Draw the whole Krebs cycle. Krebs cycle. Krebs cycle. Krebs cycle. Krebs cycle. 29. Describe the 2 steps in which decarboxylation occurs. Oxalosuccinic acid to alpha-ketoglutarate, alpha-ketoglutarate to succinyl-CoA. Oxalosuccinic acid to alpha-ketoglutarate, alpha-ketoglutarate to succinyl-CoA. Oxalosuccinic acid to alpha-ketoglutarate, alpha-ketoglutarate to succinyl-CoA. 30. Decarboxylation is catalyzed by enzyme. Decarboxylase and alpha-ketogluteric dehydrogenase complex. Decarboxylase and alpha-ketogluteric dehydrogenase complex. Decarboxylase and alpha-ketogluteric dehydrogenase complex. 31. In which reaction of Krebs cycle, GTP is synthesized? Succinyl-CoA to succinic acid. Succinyl-CoA to succinic acid. Succinyl-CoA to succinic acid. 32. How many substrate level phosphorylation happen in Krebs cycle? 1. 1. 1. 33. NAD plus H plus is released in which steps? I. Isocitrate, oxasuccinic acid. E. Alpha-ketogluteric acid, succinyl-CoA. E. Malic acid. Oxaloacetic acid. I. Isocitrate, oxasuccinic acid. E. Alpha-ketogluteric acid, succinyl-CoA. E. Malic acid. Oxaloacetic acid. I. Isocitrate, oxasuccinic acid. E. Alpha-ketogluteric acid, succinyl-CoA. E. Malic acid. Oxaloacetic acid. 34. FADH2 is released in which step? Succinic acid, fumaric acid, succinic acid, fumaric acid. Succinic acid, fumaric acid. Succinic acid, fumaric acid. 35. No of CO2 molecules released in one Krebs cycle is 2 dash 2 dash 2. 36. Write the enzymes involved in Krebs cycle. In above diagram. In above diagram. In above diagram. 37. Write the energy equivalents produced in one Krebs cycle. 3 NADH, IFADH2, IGTP. 3 NADH, IFADH2, IGTP. 3 NADH, IFADH2, IGTP. 38. Dash is the first member of the Krebs cycle. OAA, OAA, OAA. 39. Krebs cycle can be summarized as one reaction as pyruvic acid plus 4 Namibian dollars asterisk plus FAD plus 2H20 plus ADP plus pi to 3C02 plus 4 NADH plus 4H plus FADH2 plus ATP. Pyruvic acid plus 4 Namibian dollars asterisk plus FAD plus 2H20 plus ADP plus pi to 3C02 plus 4 NADH plus 4H plus FADH. Pyruvic acid plus 4 Namibian dollars asterisk plus FAD plus 2H20 plus ADP plus pi to 3C02 plus 4 NADH plus 4H plus FADH2 plus ATP. Pyruvic acid plus 4 Namibian dollars asterisk plus FAD plus 2H20 plus ADP plus Pi23C02 plus 4 NADH plus 4H plus FADH2 plus ATP. 40. What is the role of NAD in cellular respiration? It acts as an electron carrier. It acts as an electron carrier. It acts as an electron carrier. 41. Its have dash no of complexes. Name them. 4. Not 5. Names are I. NAD ubiquinone oxidorrectase. E. Succinate dehydrogenase or succinate COC reductase. E. CYT. BC1 complex or COC2 cytochrome C reductase. IV. Cytochrome C oxidase. Dash. 4. Not 5. Names are I. NAD ubiquinone oxidorrectase. E. Succinate dehydrogenase or succinate COC reductase. E. CYT. BC1 complex or COC2 cytochrome C reductase. IV. Cytochrome C oxidase. Dash. 4. Not 5. Names are I. NAD ubiquinone oxidorrectase. E. Succinate dehydrogenase or succinate COC reductase. E. CYT. BC1 complex or COC2 cytochrome C reductase. IV. Cytochrome C oxidase. 42. Electrons from complex I are passed to ubiquinone. 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 43. Reduced ubiquinone is called ubiquinol. 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 44. Aniquinone transfers electrons to dash via dash, CYTC via cytochrome BC1 complex, CYTC via cytochrome BC1 complex, CYTC via cytochrome BC1 complex. 45. Complex 4 contains 4 CYTA, CYTA3, 2 centers, CYTA, CYTA3, 2 centers, CYTA, CYTA3, 2 centers. 46. CYTC, a small protein is attached to the inner surface of the outer mitochondrial membrane. TF. F. Outer surface of the inner membrane. F. Outer surface of the inner membrane. F. Outer surface of the inner membrane. 47. Dash act as a mobile carrier for transport of electrons between dash and dash, CYTC, complex 3 and IV, CYTC, complex 3 and IV, CYTC, complex 3 and IV. Dash act as a mobile carrier for transport of electrons between dash and dash, CYTC, complex 3 and IV, CYTC, complex 3 and IV, CYTC, complex 3 and IV. 48. Write the whole ETC with each and every element is given in the NCERT diagram. It's complex 1 ubiquinone, complex 3 CYTB, Fe-S, CYTC, complex IV, Qua, Cyta, Cyta 3 cub. Complex 2 ubiquinone, complex 2 ubiquinone, complex 3 CYTB, Fe-S, C, complex IV, Qua, Cyta, Cyta 3 cub. It's complex 1 ubiquinone, complex 3 CYTB, Fe-S, CYTC, complex IV, Qua, Cyta, Cyta 3 cub. Complex 2 ubiquinone, complex 3 CYTB, Fe-S, C, complex IV, Qua, Cyta, Cyta 3 cub. It's complex 1 ubiquinone, complex 3 CYTB, Fe-S, CYTC, complex IV, Qua, Cyta, Cyta 3 cub. Complex 2 ubiquinone, complex 3 CYTB, Fe-S, C, complex IV, Qua, Cyta, Cyta 3 cub. 49. 5. Molecules of NAD give dash ATP. Dash 15. Dash 15. Dash 15. 50. Dash molecules of FADH2 give 8 ATP. 4. Dash 4. Dash 4. 51. For each ATP dash H plus passes through the 4. 4. Dash 4. Dash 4. 52. NAD pumps dash H plus into the intermembrane space. 10. According to the latest research. Dash 10. According to the latest research. Dash 10. According to the latest research. 53. FADH2 pumps dash H plus into the intermembrane space. 6. According to the latest research. Dash 6. According to the latest research. Dash 6. According to the latest research. 54. Role of oxygen is limited to the terminal stage of the process. TF. T. 55. Dash acts as a final A acceptor or H acceptor. Oxygen. 56. F1 is the integral, peripheral membrane protein complex. Peripheral. 57. Catalytic site is present in F1. 58. In aerobic respiration, one molecule of glucose produces dash ATP. 38. 59. NAD oxidation is slow in fermentation. TF. T. 60. Glycerol enters the respiratory pathway after being converted to PGAL. 61. Fatty acids enter the pathway after being converted to acetyl-CoA. 62. Amino acids enter the pathway after being converted to pyruvate, acetyl-CoA, or at any stage in the Krebs cycle. Pyruvate, acetyl-CoA, or at any stage in the Krebs cycle. Pyruvate, acetyl-CoA, or at any stage in the Krebs cycle. 63. Which biomolecule is common in the breakdown of fats, carbohydrates and protein? Acetyl-CoA. 64. Respiratory pathway is an dash pathway rather than a catabolic pathway. Amphibolic. 65. Define RQ. Volume of CO2 evolved, volume of O2 consumed. Volume of CO2 evolved, volume of O2 consumed. Volume of CO2 evolved, volume of O2 consumed. 66. RQ of carbohydrate is 1 dash 1 dash 1. 67. RQ of fats is less than 1 dash less than 1 dash less than 1. 68. RQ of trypamethane is 0.7 0.7 0.7. 69. RQ of protein is about 0.9 0.9 0.9. 70. Pure proteins or fats are never used as respiratory substrates. TF. T. 71. In anaerobic respiration, R2 equal to infinite. 72. In germinating seeds, fermentation occurs. TF. T. 73. ATP synthase is also called oxysome. 74. EMP pathway is the only process of respiration in anaerobic organisms. TF. T. 75. Distance that gases must diffuse even in large plants is not great. TF. T. 76. End product of photosynthesis is sucrose. 77. Glucose and fructose readily enter the glycolytic pathway. TF. T. 78. Glucose and fructose are phosphorylated to give glucose 6-phosphate. TF. T.